perhaps why we are not starting more punctually. So this session this morning will be dedicated to immune vascular mechanisms in these post-viral syndromes, and we will have a mix of talks discussing neurovascular changes associated with these diseases, as well as some immune mediator or immune changes in this disease. And the first speaker is Michael VanElzakker from the Tufts University, and is a neuroscientist at the Massachusetts General Hospital and Harvard Medical School, and co-founder of the Polybio Research Foundation. He will talk about his lab's research program on neuroimaging of infection-associated diseases. Michael, if you're ready, thank you.
Okay, thanks everyone. And how do I get this to go here? I don't know if the slides are up or what's the scoop. Okay, so thanks everyone for being here in the morning, and thanks so much to the organizers for all the work that you've done to pull this together and inviting me, even though I don't speak Portuguese. I appreciate it. So I'm going to talk a little bit about cognitive function and neuroimaging for neuroinflammation in ME/CFS and long COVID. So I'll sort of stretch across both conditions in this talk. So we wrote a review article a bunch of years ago that sort of got into the mechanisms and the methods of studying this topic. And so some of what I'll be talking about today is an effort to sort of make that happen. So, I think everybody knows that when long COVID came up in sort of, in the public, it took a lot of people by surprise, I think. But for a lot of people in this room, it unfortunately wasn't surprising. We sort of knew that there are consequences that can happen for some, subset of people who experience an infection where they don't fully recover. So Amy Proal and I wrote a review article at the time to try and prevent the, this is some big mystery, we don't have good leads type narrative from taking big hold. And I think it stood up pretty well. And, some of what I'll talk about today sort of gets at some of the questions that we were addressing. So this is just an overview of some of the stuff that we're doing in the lab. And basically everything except for the fibrin PET imaging we're doing on both pre-COVID, pre-2019 ME patients as well as long COVID patients. So we've done our best to sort of use the infrastructure and kind of funding bolus that came with long COVID to study people that were sick before 2019. A lot of the times it's relevant. I don't think it's the same thing. So I'll just start off by saying that. I think that there are shared mechanisms. Surely there must be, because there are shared symptoms. But I don't think it's precisely the same thing. And I think that is long COVID and pre-COVID ME. And I think that that's an important task for the field to figure out what things are similar and what things are different. And we ought to be doing sort of precision medicine in that way, I think. So I'll start off by talking a little bit about this paper here that we put out a couple of months ago. And, this is a study of neuroinflammation in long COVID patients. Essentially we tried to, get this paper out a little bit quick. So we used pre-COVID controls that were already done in the same scanner with the same basic protocol. And what we did in this project was to study a form of neuroinflammation. So microglia are the resident macrophages of the central nervous system. So when you look at a brain, a significant number of the cells you're looking at are microglia or other forms of glia, which are neuroimmune cells. Glia means glue in Greek, sort of a pejorative where they thought, originally that they were just scaffolding for neurons, but it turns out that they're much more important than that. And they're innate immune cells. So when they come in contact with pathogen-associated molecular patterns, whether it's a full pathogen or an antigen, or when they come across neuroimmune mediators such as pro-inflammatory cytokines, they actually activate, they change form, and they sort of withdraw their arms, and they take on this sort of thicker form, and they release a bunch of mediators. That includes glutamate itself, which is, of course, the main neuroexcitatory neurotransmitter, as well as other pro-inflammatory cytokines, all kinds of things, prostaglandins, ATP, bradykinins that excite neurons. This, of course, can disrupt the normal sensitive signal-to-noise ratio of neuronal signaling. It's important in the short term, but at the long term, it can become problematic. And it also releases what's called the translocator protein, or TSPO. And this is what we're able to measure with PET neuroimaging. To be fair, immune cells, peripheral immune cells, also upregulate the TSPO protein. And so the signal that we see in the brain can and probably is sometime immune cells that are penetrating in from the periphery. So in this study, we were looking at 12 adults with long COVID, mostly non-hospitalized, mostly pre-Omicron, mostly pre-vaccine. And these are patients that qualified for a modified ICC criteria for the MEC. Have someone with long COVID and that's about it. That's possible to have, right? So we were looking at this sort of sickness syndromic manifestation, and we have additional controls going, but this is 43 controls from the scanner before COVID. So sort of a standard PET protocol using what's called the PBR28 radioligand. The translocator protein used to be called the peripheral benzodiazepine receptor, so PBR. So this is a radioligand that binds to that protein that we're interested in. And as it does, because it's radioactive with really rapid decay, it will release protons right there where it's detecting the neuroinflammation, and we can localize that. And this is just some statistics of the PET analysis. So this is sort of what our results look like. And this is a statistical map showing the regions of cerebellum normalized signal that were greater in the long COVID patients than in our 43 pre-COVID controls. We extracted a whole brain signal, and this is what the whole brain signal looked like. I put the two you probably can't see it, but I put the two hospitalized patients in open circles just to show that it wasn't the case that those were the two most extreme patients. So you see kind of a diversity of signal in the patients and in the controls, but a pretty solid split between the cases and controls. We wanted to see if this was driven by a particular area. So these are sort of the brain regions that were activated in our long COVID patients greater than controls. We kind of saw that the signal was somewhat associated with areas where there's a bit of a window in the blood-brain barrier. So for example, at the roof of the ventricles, right below the corpus callosum, there's a little bit of a window in the blood-brain barrier, or circumventricular organs. I wouldn't want to overstate it, but we sort of saw a signal that was consistent with that. So we extracted data from the different regions, and it didn't seem like one region was really driving the effect. So it did seem to be sort of all over those regions. This is the just as an example region, this is the subfornical organ. So it's one of the 7 circumventricular organs that naturally, normally in everyone, has a diminished blood-brain barrier. The circumventricular organs are really rich with ACE2 receptors. And so we thought maybe that was part of the story, and maybe that mattered. Here it is, it's number 3. And so we thought, okay, if we're getting signal here, then maybe it's the case that there's, something in the blood that's at least contributing to this, because this is the place where the brain sort of communicates with the blood, or one of the places. And so we did a bunch of measurements of blood that we took immediately before the people got into the scanner. And we found that 6 different mediators correlated pretty well. I think it was Massimo yesterday that was saying they only had a 0.7 correlation. Usually in biology, that's really good. And sort of surprising. So we found a 0.7, moderate to strong correlations. For example, this one up here on the left is SL-selectin. So that's an adhesion molecule, meaning that it is expressed on the vasculature so that it pulls in immune cells that are rolling through the blood. They attach to the vasculature and get drawn in. So that's where that, assertion that our signal may be penetrating neutrophils as well as the resident macrophages or other glia that are in the brain. And fibrinogen, of course, the Riccia pretorius group, works on this cascade, and we found that it correlated at a 0.8 with the whole brain neuroinflammation signal. So our next step that we did was to use COVID recovered controls. So these are all people that had COVID and report feeling fully recovered. And this is showing signal that's significant in the brainstem. So the signal location did sort of move downward, but it's still what we would call subcortical, sort of a little bit of a diminished blood-brain barrier. It's not the case that I can just look at a scan and go, there's the long COVID patient. The neuroinflammation is there. There's a split where in our data as of now, the long COVID patients are largely at the mean of the controls or above, but you really can't just look at a scan and diagnose. And I think that's true in these diagnostic labels of long COVID and ME/CFS and the infection-associated chronic illness. There are, at the group statistic level, these biological factors emerge, but it's really not just a biomarker that you can just look at and say there it is. So we also have, even before COVID, we tried to have this study sort of rolling and we got it shut down, but then back up again. So we have pre-2018 ME patients, which obviously onset was not COVID-19. And we found a signal as well in a region that we predicted ahead of time called the anterior mid-cingulate. So cingulate gyrus goes right above the corpus callosum there. It means belt in Greek. It wraps around the corpus callosum like a belt. And I'll come back to this brain region because I think it's important for the symptom of brain fog. Again, you can't really look at the scan. If you look over at the data, you can't really just look at the scan and say, there's the ME/CFS, but it really is the case that it splits out at a group level. And we also found brainstem signal there. I think this is where the idea that maybe not the same thing is driving, but there are shared pathways that lead to shared symptoms is kind of how I think about it, right? So there are probably aspects that you can sort of attack across patient groups if you're going to manage symptoms. But if it's really being driven by antigen, or if that's a component, you have to address the antigens. That is a specific approach as opposed to a non-specific approach. This signal, by the way, is a little bit more similar to what Nakatomi found in 2014 with an earlier radioligand and what Rodmakers found. Rodmakers did not find in 2021. And they did a good job in their study, but they were using patients that had been sick for a really long time. It was all women and all sick from Q fever. So those may be some of the reasons that they did not replicate what other groups have found. So I want to talk a little bit about the symptom of brain fog and the idea that these are overlapping symptoms across the different drivers. This is a really common non-specific symptom that exists in all kinds of things. Almost everything really has at least some aspect of brain fog in some patients. So the way that we approach this is to try and get at a really fundamental aspect of cognition, which is called top-down cognitive control. So the idea is that if you were looking for a friend in a crowd who was wearing a red shirt, you would be focused, you have a goal, and you're ignoring extraneous things on purpose, and you're focusing in on something on purpose. That's called stimulus-driven or top-down cognitive control. And it's a really fundamental aspect of cognition. So when I'm searching my mind for someone's name or phone number, it's largely the same circuitry. So there's my friend with the red shirt that I found. But if suddenly a red balloon floats up, I wasn't looking for that, but it's going to capture my attention whether I like it or not. That's called bottom-up attentional capture, and it's actually a different circuit. And it's an important part of cognitive control, is to maintain focus on what you're doing while you're ignoring things that are happening around that are not important to you. And I think that's a lot of what the patients kind of deal with, is a struggle. Lights and sounds and those sorts of things are really sort of distracting. There's a sensory component there too, but some of it is cognitive as well, I think. So when that happens, you sort of lose focus on the thing that you care about and end up having your attention grasped by the thing that you weren't actually focusing on. Well, it turns out that that's largely held in place, cognitive control, by that region that I mentioned earlier called the anterior midcingulate. And this is a meta-analysis, so pre-COVID meta-analysis, just studying the effects per se of inflammation on cognition. So this is a combination of 24 different studies, 400 plus patients who either had interferons injected or lipopolysaccharide, or even just had blood extracted, and they measured inflammation. And up there at the top is that anterior midcingulate. It's part of the sickness circuitry. So it's an area that's affected by inflammation. It turns out that, and that's that anterior midcingulate. It turns out that you can study it really directly with a task. So this is a task that was developed by my PhD mentor, Lisa Shin. It's a really interesting brain region because it's both important for cognitive control and sensitive to inflammation. So the task that can address this region is called the multi-source interference task. And it reliably activates what's called the top-down attentional circuit that sort of holds control despite the fact that other things are happening in your periphery. Whether it's periphery, literally in your visual field, or cognitively as well. So we used a dual PET MR scanner, even for the data that I've showed you already. They were in an MRI scanner that has a PET insert, which allows us to do multiple forms of neuroimaging at the same time. So we're able to do an fMRI task while we're collecting the PET neuroinflammation data. So the multi-source interference task, like other tasks in fMRI, we have a little MRI safe joystick basically that's down there in the lower right. And the way that it works is you place your fingers on it, 1, 2, 3. So if you kind of put your hand up with your palm facing me, 1, 2, 3, that's where the button box, the numbers on the button box are. And the way that the task works is you pick the numeral that is different than the other numerals. So for this one, you would pick 1, right? For this, you'd pick 2. For this, you'd pick 3. For this, you'd pick 3. Now that's hard, right? That's the interference condition, because your finger position wants you to choose your number 1 finger, right? So you have to override that. You have to override what's called the prepotent response. And this is a real reductionist way of getting at that mechanism of cognitive control. It taps into that same circuit that we care about. So let me just give it to you if you want to put your hands up at about the pace that they get in the scanner. So that's 3, 2. You can see how much easier it is when it's like that. So, and what we do is we compare the non-interference to the interference And everybody, healthy or not, will have more activation of this circuitry in the interference condition. And the question is whether patient groups need more real estate. They need more brain power to complete the task. And it turns out that yes, they do. So this is long COVID patients. Essentially, it's a pretty good split in the data where they're able to complete the task. It just takes more brain effort, essentially, to complete the task. And so what I've done next is to show you an overlay. So this is that MECFS group pre-2018. This is the anterior mid-cingulate of, in this case, I'm showing you 9 controls versus 7 patients. And that's that anterior mid-cingulate. And now I've overlaid, if you can see the blue, that's the task. That's where the patients, in this case, MECFS patients, needed more activity to do the task than the controls did. So we're going to put together the full story. This is sort of showing the concept that we're working to get at. So what we're doing here is showing you that it's really that same region that's affected by inflammation that's important for cognitive control that is affected. It really overlays really well. And that's that anterior mid-cingulate. So another thing that we found from another ongoing study, this is a study, a separate study that uses EEG as well as fMRI in patients, again, long COVID and then pre-2018 MECFS patients. Here, I've combined the patient groups, again, not because I think they have the exact same thing happening to them, but I think the symptom expressions are the same. And what we're seeing here is delayed phase index. So that area of anterior mid-cingulate ought to be kind of pulsing with activity at the same rate as a region further back called the superior parietal that holds cognitive control as people are holding a task. They ought to be sort of turning on at the same time. The brain waves ought to be aligned. And this is showing what's called the phase lag, which is sort of a common thing that can happen in problems with cognition. And you're seeing basically a delayed phase in the MECFS combined with long COVID patients. We'll split it out. This is like hot off the press type data that we're in the middle of. And interestingly, that lag anti-correlated with the MCIT task performance. So in other words, they did sort of worse on the task, moderate correlation, 0.45, but significant. So it's the task performance. Okay, so that's anterior mid-cingulate. I'm also interested in, oh, sorry, I forwarded, this is that correlation, the anti-correlation between the phase lag and the MCIT task performance. So we're also interested in the brainstem as a region we think is important in these conditions. So if you look at that meta-analysis of regions that are affected by inflammation kind of per se, brainstem is all the way over there on the left. And it kind of makes sense. This is where the vagus nerve enters the central nervous system. So vagus shares the same Latin root as vagabond because it wanders all over the body. It detects small amounts of inflammation. This is why you can be sick in the lower left lobe of your lung and have a full body response, despite the fact that cytokines are, basically kind of mid-sized proteins that don't easily cross the blood-brain barrier. So the vagus nerve acts as a diffuse sensory organ that detects inflammatory mediators and signals, hey, brain, the body is sick. So for example, if you inject a rat with something like lipopolysaccharide, broken up bacterial cell walls, the rat will act sick. It won't socialize, it won't run on a wheel unless you have cut the vagus nerve. And then the brain doesn't know that the body is sick. There are still some aspects of the sickness response that will get communicated, but some won't. And so, we're real interested in whether the brainstem can be sort of an on-ramp where, different forms of stimulation provocation, including physical compression with CCI patients, might be driving symptoms that overlap. Again, that drives the sickness response that makes people sort of feel really tired and worn out and in bed. And I guess let me just take just a real quick second here. The sorry, let me get here. The NTS here over on the right is the nucleus of the solitary tract, which is where the vagus nerve enters brainstem. The nucleus ambiguous just below is an outgoing nucleus that, for example, problems there would lead someone to be vulnerable to tachycardia. Ventrolateral medulla and dorsal motor vagus are outgoing autonomic control regions. So problems there would lead to problems with autonomic control. And then the DRT down below is the dorsal reticular nucleus, which receives from the periaqueductal gray, that's inflammatory pain. And so problems there, sensitization would lead to an increased sense of pain. And that circuit, by the way, is sexually differentiated, where it's more sensitive in women. So what we did was to extract data from the brainstem using magnetic resonance spectroscopy, which allows us to look at the chemical content of brain tissue. And we found that in both the long COVID and the pre-2018 ME/CFS patients, there was reduced N-acetyl aspartate, which is a marker of sort of metabolic health in brain tissue. Again, I can't just look and say there's the patient, but this is where sort of dysfunction happens. And let me just go kind of real quick through the next couple of slides. So this is hopefully an animation. Yeah. So what we did is to take live neutrophils. We didn't freeze this blood. The cells are still alive, and they're swimming into a little well to get at bacteria. And what we found was that the ME/CFS patients had a higher proportion of cells entering the wells than the controls. The long COVID is just a trend at this level, although it's probably underpowered. So evidence of immune activation. Again, that's the innate immune system, which means that there's a shared response despite potentially non-shared drivers. And then we gave our participants a wearable sleep device to wear the 2 nights before they came in for the scan. And as you might guess, found reduced sleep quality in the patients versus controls. This is a measure of fragmentation and periodicity. We also do pupillometry, where we, in a darkened room, sort of put this device up to someone's eyeball, measure the diameter of the pupil, and then flash a quick light to see the contraction. And in this case, the long COVID patients have a reduced initial diameter relative to both the ME/CFS and the long COVID groups. So this is one area where the long COVID and the ME/CFS are not the same. And also the end diameter after the constriction. This is an area where the ME patients are different than both groups, which is constriction velocity. The velocity here is sped up in the pre-2018 ME/CFS patients, a little bit like a concussion patient. And this is the max constriction velocity. So I just want to thank my lab, who does a great job. The patients are really generous in coming in. We're not even trying to get them better, but they all report having a good experience. And that has a lot to do with my team. So thank you very much.